Hello, thank you for tuning in to our online lecture on our excessively cold day. Today we're going to be talking about chemical reactions. So on Monday, we went over the basics of some of like atoms and chemical bonds and ions and just the basics of some chemistry to make sure that everybody's on the same page. Today we're going to talk about chemical reactions in general and talk about enzymes and sort of get on board with the real chemistry that we're going to be dealing with in this semester. So to talk about chemical reactions, we have to talk about energy and types of energy and what we're looking at here. So when we think about movement and force in the physical world at the scale we interact with, a lot of what we see is kinetic and potential energy. Kinetic energy is movement, and potential energy is stored energy. So an example that we're seeing on this screen here, we have a bicyclist who's putting in energy to get up to the top of the hill. At the top of the hill, they have stored potential energy from gravity. And then as they go down the hill, they're turning that potential energy back into kinetic energy to move. Chemical reactions, we're going to see a lot of very similar processes. Now we have a lot of different types of energy. There's radiant energy that comes, so from the sun is the most common source that we think of. When we think about radiant energy, energy that's radiating from a body out to other things. We have electrical energy, which we use day to day in our daily lives, but isn't something we think about in this way too much in biology, although we did, of course, talk about electrical currents in the neurons, and that's something that's important. Chemical energy is what we're going to be talking about a lot here. We've talked about ATP as the energy currency of the cell. And this is where we're really going to get into what does that mean? What does it mean for ATP to store energy, to give off energy, those sorts of things. We also have sound, thermal energy, which is heat, and that's also going to be very important, both as for sort of setting a base energy level for biological things, and also waste heat, waste energy that is released when pretty much anything else happens. Whenever forms of energy are converted, you get waste heat released. Nuclear, we're not going to deal with too much, although we have talked about it a little bit in terms of radiation. Mechanical, we're going to deal with in terms of what we produce. When our muscles use energy to contract, they're turning chemical energy into mechanical energy. And magnetic, we're really not going to deal with in this class. So energy is constantly being converted from one type of energy to different types of energy. So a tree, when we're looking at a plant, it converts solar radiant energy into carbohydrates, which are a form of stored chemical energy. Then that stored chemical energy can be consumed by an animal. It's turned into glucose, which is, so we break down starches. We'll talk a lot more about this in the digestive section, and other complex carbohydrates turn that into glucose, which is still stored chemical energy. Then that chemical energy is transferred to ATP, where it can be utilized directly. And here we're looking at muscles, where it's converting that chemical energy of glucose and then ATP into mechanical energy to move, with heat flow as a waste product. Now, mammals do also sometimes use heat as the goal, so we may directly be going for heat as the product, but heat is always going to be a waste product whenever you're converting energy. So this is an effect, and we sort of utilize that waste product to maintain a stable temperature. We'll talk a lot more about that later in the semester. So when we talk about chemical energy, we're talking about how much energy is stored in the bonds of a particular chemical. When we add energy to those bonds and store energy, that is an endergonic reaction. There is less energy in the stuff you started with, the bonds of the stuff you started with, than there is in the bonds of the stuff you finished with. It means you've stored energy in that process. Then there are exergonic reactions, which are just going the opposite direction. So you're taking something that has more stored energy, releasing that energy, and getting out something that has less stored energy. But something you'll hopefully notice on here is that there's a point on each of these graphs that is higher than either the starting point or the final point, either the reactants or the product. You have a point that's higher. That difference between the starting point and that highest point is the activation energy. And that much energy needs to be put into the reaction to make it happen. In exergonic reactions, you're going to get that energy back, but you still need to put the energy in to make it happen. And that's going to be very important when we get to enzymes in a little bit. When we talk about chemical reactions in general, we can talk about things coming together. So you've got two reactants that come together to form one product, things breaking apart. One reactant breaks apart to form two products. Things can swap around. So we might have, in our third example here, we have B swapping with C, or B and D swap, or C and D swapping with B. So, or C swapping with D on A and B. So there's lots of different ways that these chemical reactions can happen. All of these are important in our bodies. In general, reaction rates, how fast these reactions happen, is always going to increase with the temperature, because the temperature is a measure of sort of how much energy there is at base. So remember, we have to get over that activation energy, and the higher the temperature is, the easier it is to get over that activation energy. It's also going to increase with concentration, simply because there's more things around to bump into. And so how fast a reaction happens is really important in our bodies. There are lots of reactions that would happen by default, exergonic reactions that produce energy that don't need a lot of work to happen, but are going to happen very slowly at room temperature with nothing else pushing them. And that is where we get into enzymes. So enzymes are catalysts. They are proteins in our body that catalyze reactions, which means that they make it easier for those reactions to happen by decreasing the activation energy. So all they're doing is lowering that hump. This is really important. They're not changing the substrate, they're not changing the product, and at the end, they're released. The enzyme goes floating off, but they decrease that difference in the state. Now, what that means is that enzymes are only helpful if the reaction would happen without the enzyme, just a lot slower. So they need to be exergonic reactions, reactions that release energy that don't need an additional energy source. Now, enzymes can sometimes allow individual endergonic reactions to happen by combining another energy source to make the overall reaction exergonic. Oh, oops, I already answered this question. That's okay. But, this is what we're talking about with, these reaction rates and these enzymes. So we're going to spend a lot more time on enzymes on the POGL. If you had a chance to look at the activity I posted, that is what we're going to spend a lot of time thinking about: what enzymes do, how they work, how they're affected by conditions in the body. So I'm not going to spend a lot of time on them right now. You'll be working on that activity later. Another really important in our body's chemistry, as has come up on Monday, is water. So water is important for all sorts of things in the body. It regulates our body temperature, protects our organs, helps the flow of nutrients throughout the body and the flow of food within the body. It helps dissolve things and make them accessible. It lubricates joints. It moves all sorts of things around the body. It's also involved in a lot of actual chemical reactions that happen in our body. But it's sort of the thing we think of it most in terms of the body is as a solvent. So water, as we talked about on Monday, is really good at dissolving salts and small polar molecules. The smaller these particles are, the better dissolved they'll be. If you have larger molecules that could otherwise dissolve in water, you wind up with either a colloidal solution or a suspension. A colloidal solution means that these are partially dissolved, but still affecting the solution. Sort of the classic example of that is Jello. Think about how Jello is clear, it doesn't become cloudy, you don't see particles in it, but that thickness, that gooiness, that's a colloid. Your blood also is in part a colloidal solution with large proteins, and that is why your blood is viscous. If you think about the feeling of blood, it's got that viscous feeling to it. That's from that colloidal effect. And then there are also suspensions, particles that are too large to be even partially dissolved, but are floating around in the water. Milk is an example of a suspension, and that's why it's white. Your blood is also a suspension in that it has large particles floating around, as well as those partially dissolved colloids and completely dissolved salts. In addition to that solvent, as I said, water is really important in a lot of chemistry and chemical reactions that happen in our body. When we make larger molecules, we often do so by dehydration synthesis. That means that we combine two molecules which have an H at one end and an OH at the other end, take out the H and the OH as water, and then that allows us to combine those two into a larger molecule. This is important for proteins, for fats, for some carbohydrates, for DNA. So this is something you're going to see a lot. On the other side of it, we have hydrolysis, where we add water to separate two molecules. This is when we need to digest those things, and to break those proteins down into amino acids, for example. Hydrolysis is how we do that. We add the water, and that allows us to separate those molecules. In lab on Monday, of course, we spent a lot of time talking about acids and bases. And we're going to come back and address those a little bit more. So hopefully from lab you remember that acids are things that release a proton, an H+ ion. Bases absorb a proton, an H+ ion. In general, in biology, we're going to be seeing bases as things that release an OH ion, because that bonds with H+ to make water. There can also be bases that just absorb H+ strongly enough that they are going to take it away from water, leaving an OH ion floating around as the remainder of the water. We also talked in lab about buffers. This is our classic carbonic acid bicarbonate buffer that is in our systems. This is our primary buffer in the blood, and as you hopefully remember from lab, buffers buffer the effect of acids and bases. They limit the effect of acids and bases. So that allows our body to absorb the effects of lactic acid production by the muscles, changing levels of carbon dioxide, things like that, without having our blood pH change very much. Finally, so we've talked about water, we've talked about acids and bases. I also want to talk a little bit about carbon. We talked about carbon a little bit on Monday, but carbon is really the center of biological chemistry. Organic chemistry revolves around carbon. Organic just means that there's carbon containing, the chemistry of carbon containing molecules. So carbon is really important because it has 4 electrons in its outer shell, which means that it can form 4 bonds. These bonds allow it to create, being able to form 4 bonds allows it to create all sorts of complicated structures, with single bonds and double bonds to other carbons, to oxygens, to hydrogens, allowing us to create these complicated structures like carbohydrates. All of our biological molecules that we're going to be talking about are complex structures of multiple carbons. Carbohydrates are made of these carbon and oxygen rings, 5 or 6 carbons, depending on what you're looking at. And these monomers are single rings. So glucose, for example, is a single 6-carbon ring. And then we also have dimers, like sucrose and lactose, that are made of 2 rings, and depending on what dimer you're looking at, it might be all the same ring, or it might, same monomer, it might be 2 different monomers. And then we can also have larger, more complex carbohydrates like starch, where you have a polymer of these ring monomers. So you've got a bunch of different ones all connected, and if you remember, those connections are made by that dehydration reaction that we talked about. Another important molecule that we're going to talk about a lot is triglycerides. These are our standard fats. In this case, we can have saturated or unsaturated fats. Saturated means that there are no double bonds between the carbons, so each carbon is connected to another carbon by 1 covalent bond. In an unsaturated fatty acid, it would be connected, some of those carbons are connected by 2 covalent bonds. Double bonds are very common and important between carbons. In organic chemistry, you'll also see double bonds like this between the carbon and the oxygen. And those are important for setting up the structure. As you can see, in this unsaturated fat, these double bonds result in different angles that those tails are bent at, which is important for the structures of those fats. These tails also show up in the phospholipids. So phospholipids have fatty acid tails, and they can have kinks in them from double bonds, just in the same way triglycerides do. And they have phosphorus and oxygen in the head that results in this hydrophilic head that is polar and therefore attracted to water, and the hydrophobic non-polar tail that moves away from water. Another non-polar fat-related set of chemicals that we're going to be dealing with are steroids. So steroids have these multiple ring structures. They're non-polar molecules. And all of these are derived from cholesterol, but they have a variety of functions in the body. Particularly, we see these a lot in hormones. So when we get to talking about the endocrine system, we're going to spend a lot of time with steroids. The next major group of organic molecules that we need to talk about are proteins. Proteins are made of amino acids. So amino acids all have this base 3-carbon structure with the OH coming off the side, and the nitrogen here. This nitrogen is really key. We talk about things being nitrogen limited, that is protein limiting. That nitrogen is absolutely key to this protein and the biggest place that we're seeing nitrogen in the body. And then there's some group coming off that it makes up what makes this particular amino acid distinctive. So there's a lot of different amino acids. I don't want you to worry too much about all of these different amino acids, but what's important about this is these amino acids come together to make up peptides. So they've got these distinctive R groups coming off. They're bonded together with those nitrogen groups. And once again, this is formed by a dehydration synthesis reaction, the same as we were talking about before. And we get these long chains of peptides. And these long chains of peptides come together to make proteins. Now, a protein can be one long chain of peptides, or it can be multiple long chains of peptides. But in addition to all of these, so one single peptide is a chain of amino acids. It can be as long as you want. Some proteins have many of them. And each chain is going to be folded with certain secondary structures. So it might have alpha helices where it forms a helix or sheets. You can also just have long straight pieces. And then those structures come together to form a tertiary structure, these three-dimensional structures. And these structures are really important to the functions of these proteins. Just like every other time we've talked about structure and function, they're closely related, and these structures are very important to how these proteins work. Well, you'll see that a lot when we get to the enzymes activity, and we're talking about the structure and the function of enzymes. In addition to the tertiary structure, sometimes you can have quaternary structures where multiple peptides come together. So in this particular molecule, we're looking at 4 separate proteins, but this could be 2, this could be 3, this could be 4. There's a wide variety of numbers. We often see the hemoglobin, which has 4 chains. That's a very common one that we look at in terms of quaternary structure. And these structures are very closely related to what happens, what the proteins do. So if you look here at myosin, we have an actin binding region that binds to actin. We have a regulatory region that is activated. We have an ATP binding site, and you can see it's kind of long and stretched out. So that's important for providing that movement along the actin, where it's crawling along actin. Myoglobin, on the other hand, you'll see is clumped together, and it has iron here that allows it to hold oxygen. So it's clumped together in a way that it can hold oxygen in the center there. These structures, you're going to see that they are directly related to that function. It's particularly important when we're talking about those enzymes. Our final category of molecules that we're going to be dealing with are nucleic acids. Nucleic acids are your DNA and your RNA. These have a sugar base with a phosphate, sorry, a sugar ring with a phosphate, and then they have some base coming off of the side, and there's a different one for A, C, G, and T. We get long chains of those sugar rings with the bases coming off the side. And this structure, of course, is important because in DNA, you get 2 long chains of these with the bases coming off the side, and 2 of them come together, and those bases form hydrogen bonds between them. But G always bonds with C, T always bonds with A. And that's important because it allows us to repair and to create new DNA by pairing up the correct bases on one strand with the ones that are already there on the other strand. And once this is all established and set, this forms this double helix shape where they curl around each other. In the other nucleic acid that we have is ATP, which I keep coming back to. So ATP is this sugar base, this sugar ring with a base coming off of it. And then on the other side of the sugar ring, there are 3 phosphate groups. And this is a high energy bond, which means that we can break off using water here, the same as we have with everything else, one of these phosphate groups, which gives us releases a lot of energy, and we can use that energy to do things, and that leaves us with ADP, which we can later rephosphorylate, add the phosphate group back on, using energy to store energy to do it all again. So this is our energy currency, is breaking up off that phosphate group. Sometimes we go down to AMP by breaking off 2 phosphate groups in specific circumstances. That AMP can also often be a signaling molecule inside the cell. We'll talk about that more when we get to the endocrine system. All right, so that is the basics, the super basics of organic chemistry at the level I need you to know it for this class. Obviously, there's an awful lot that we haven't covered. I know I'm not there to take questions right now, but I will be, as I said, online the whole time that we would normally have class, as well as during my normal office hours. So I'm happy to take questions by email or by Google Hangouts on the email that I've posted. I wanted to go over the POGL a little bit for those of you who haven't done a POGL before. Now I'm hoping that despite the fact that we don't have class, that I want to encourage you to get together online and do the POGL together. I have posted this as a group activity. Now it is very important that you put the names of everyone who participates when you submit this, because since we're not in class, I am not observing, so I don't want people to not get credit for work that they did do, or to get credit for work they weren't participating in. So please write down everybody who was involved. Now in POGL activities, this is a little harder online, but I try and get everybody to take on an individual role. We'll be doing a bunch of these. The POGL stands for Process-Oriented Guided Inquiry Learning. So on the first day, I talked about discovery learning, and this is one of my favorites for discovery learning, for working through some of the really hard concepts like enzymes and how they interact, and figuring it out yourself. Now, normally I try and check in with each group after, at various points. You'll see in the activity it has stop signs at particular points. Obviously, it's not fair to me to make you email me and wait for my response, but I am very happy to talk to you about these things, and I would encourage groups, if at all possible, to check in with me on things before you turn it in. It's not a requirement, but I do encourage it. So in these POGLs, there are roles. This may be a little hard to do in an online environment, but I encourage you to give it a try. You can modify the roles as you need. So these roles can change from POGL to POGL or stay the same as long as everybody is happy and participating. Do what your roles call for, but feel free to engage in other ways as well. As I said, normally I'd have you check with me after every stop sign. It's not really practical in an online environment. So the roles for POGLs, we have the reader. So the reader's job is to read each piece out loud. This is not an activity where you should divide up the work. Sometimes that's fine. These ones, that defeats the purpose. The whole part is working through it and figuring it out together. So the reader reads each piece out loud to make sure everyone's staying together. The recorder writes down your group conclusions. So when your group has agreed on something, the recorder writes it down. The manager's job is to keep the group on task and make sure everyone fulfills their roles and to ensure that everyone is heard. This person is not just going to jump ahead and lead by doing. This person is here to make sure that everybody is on task and everybody's talking it all over. The quality control person is there to question your conclusions, to really ask, are we happy with that answer? Is that good enough? Do we really understand this? That person is one of the most important roles here, just to really make sure that everything is up to snuff. And then here there's the spokesperson, but I guess that's not actually relevant here. So just make sure that these ones are covered if at all possible. And that brings us to the end of the recorded lecture. As I said, I'll be available by email or by Google Hangouts. Please contact me if you have any questions. And the POGIL is due at 11:59 p.m. Wednesday night. So you've got all day to ask me questions if there's anything you're struggling with. I hope everything goes well, and I will see you on Monday.